And naturally, we engage in glycolysis when we have a lot of energy at our disposal. And we might engage in the process called gluconeogenesis, which forms glucose when we don't have a lot of energy at our disposal. But one thing I just want to kind of wrap up glycolysis with, and we'll, after today, I think the next lecture after this, we'll be focusing on the breakdown of glycogen to provide a source of glucose when we need to rely on our energy stores. Actually, that's dietary glycogen over here, but endogenous glycogen would be our stored glycogen, and dietary glycogen or starch could be immediately broken down to glucose. We'll see that endogenous glycogen actually, through the activities of certain enzymes, gives us glucose 6-phosphate. It initially gives us glucose 1-phosphate, and then that's mutased to glucose 6-phosphate. This is kind of a complex figure, so I think that the main points here is in terms of glycolysis, we're getting our sources of glucose from diet or from stored sources. We can also get it from sources like sucrose in our diet. But I'm not going to try to trace every single pathway that's shown here. I think it would be counterproductive. So let's move on to pyruvate, which was the final product that we got from the glycolytic pathway. And we're going to see what can happen to it. And we're going to return to something we did discuss the last time, which is pyruvate under anaerobic conditions or anaerobic conditions. We did go over this the last time, but just by way of review, I'm going to remind you that under anaerobic conditions, we can get lactate or, depending on the organism, ethanol. We're going to go into that in more detail today. And under aerobic conditions, pyruvate is converted to acetyl-CoA, and that goes into the citric acid cycle, which is a few lectures from now. Okay, so let's take a look at what happens when we have either lactic acid fermentation or ethanol fermentation. So lactic acid, the formation of lactate can take place in our own bodies, but it also takes place in microorganisms. One of the things that it does in the process of converting pyruvate to lactate is it actually oxidizes NADH, right? So NADH here is converted to NAD+. That is an oxidation reaction. And then pyruvate is actually reduced to lactate. So it's the lactate that's gaining the electrons, and the NADH is giving up the electrons. The enzyme in question here is lactate dehydrogenase. So one thing we need to consider is that for glycolysis to go forward, we need to maintain a certain level of NAD+, right? Because glycolysis is in the business of taking NAD+ and making NADH. But as you can imagine, there's always competing demands, right? Some NADH goes off eventually and is carried to oxidative phosphorylation because it is an electron carrier. But if this never happened, if we never regenerated any NAD+, glycolysis would come to a screeching halt. Because if we look at glycolysis, there is a redox reaction in the payoff phase where this happens, right? So we need some anaerobic conversion of pyruvate in order to maintain stores of NAD+. Okay. The lactate itself can actually be recycled. Recycled to what? It can be turned into glucose. So, so if we go from glycolysis to pyruvate to lactate, like lactate, we can just do the whole thing in reverse and we can get back to glucose. As you can imagine, if we're performing strenuous exercise, anaerobic conditions start to take over, and we produce a lot of lactate. And in a very sudden and super physical and intense kind of exercise, the reason this all comes, there's only so long we can maintain this kind of thing, right? And one of the reasons that this can only go on for so long is that the lactic acid will ionize, and this is going to acidify conditions in our bodies, and it will then say, okay, enough, can't do this anymore. Can't do this anymore. We know acidification of the blood has a lot of implications, right? And so yeah, so it can only go on for so long. Intense physical activity for these reasons. Okay, what about ethanol? Well, last time I checked, we cannot make ethanol in our own bodies. We can consume it, and sometimes detrimentally, but sometimes socially, very pleasant. So what organisms can make ethanol? Well, a big one is yeast. Yeast is used in the making of beer and wine. And this also regenerates NAD+, but it does it in a different way than lactate fermentation. It actually takes pyruvate and it decarboxylates it. So notice in this case, we're going from a 3-carbon molecule ultimately to a 2-carbon ethanol. Well, right away we go to a 2-carbon molecule, which is acetaldehyde. So CO2 is released at this point. It's worthwhile taking note of the cofactors that are at play here. TPP, which is something we'll address in more detail later, is one of the cofactors, and magnesium ion is the other. Then after acetaldehyde is formed, we do have this redox reaction where once again, NAD is oxidized and acetaldehyde is reduced to ethanol. So a 2-step reaction here, as opposed to the 1-step conversion of pyruvate to lactate in the other form of anaerobic fermentation. So you don't really have to know these mechanisms, but it's kind of interesting maybe to take a look at at reaction number 2. By the way, TPP stands for thiamine pyrophosphate. So it's another one of these coenzymes that has a B vitamin as its precursor. We will take you don't have to know this mechanism, but it's kind of interesting to see that it's NADH that does the nucleophilic attack on this carbonyl carbon of acetaldehyde. And if I had a nickel for every time we saw this, right? I mean, every time it's a variation, but so often we're seeing in these bioorganic reactions, attack at a carbonyl carbon, and then ensuing events. This time we have a ring at play. So there's some interesting chemistry going on here with, the arrow pushing. And another thing to take notice of, I mean, this is a less common, this looks less like a nucleophile than some things that we've seen previously. Like when you see an alkoxide ion, you say, okay, oh boy, that's really nucleophilic, right? Here, we require the zinc coming into play here in order to polarize this carbonyl group here to really make this reaction possible, and to get the transfer of a hydride ion onto the acetaldehyde and form the ethanol. So kind of an interesting reaction, I would say. One thing that's fun to take note of is, ethanol, of course, is mentioned, beer, wine, what else? Well, no, I'm trying to think. No, I'm thinking of yeast and bread, but that's a whole, huh? Well, for lactic here, yes, for lactic acid, not ethanolic fermentation, but for lactic acid fermentation, yes. Look at all these examples. We have pickles, yay. Sauerkraut, sausage, weirdly, soy sauce, definitely fermented. I don't normally think of sausage as being fermented, but maybe certain kinds of sausage, I don't know. Kimchi, kefir, dahi. Does anyone know what dahi is? Yogurt. It's a kind of yogurt, okay. Is it, what country are we? Huh? Oh, okay. And kombucha, never touched the stuff personally, but some people do. So what is going on here with this? I mean, why is it? It's tasty for one thing, we know that, right? I mean, a lot of these things are enjoyable to eat, but it also serves a purpose insofar as the drop in pH preserves the food from spoilage. So, we normally store these things in the refrigerator even so, but we know pickles, like, I mean, I've had pickles in my refrigerator for, it's not like other things you put in the refrigerator and mold and spoil and whatever. Pickles will pretty much last forever, right? So anyone here ever made yogurt? My mom and I used to make yogurt when I was a kid, and it was like we'd see these things on TV and it would be like the yogurt maker, which you really don't need whatsoever to make yogurt. But all it really does is I'm pretty sure all that, first of all, it had the little cups, right? It had these little cup-sized things for you to make your yogurt in. And it gave you some kind of powder, I'm sure, that would be some kind of starter thing that had the bacteria in it, this lactobacillus bulgaricus, whatever. And it would maintain it at a certain temperature, just a little higher than room temperature, right? And that would kick. So it would do it a little faster than it would if you just let it sit around at room temperature. But you could really do it without. Anyway, we were very proud of ourselves. We made yogurt. Okay. So now on to gluconeogenesis. If there was ever something that, I mean, they both sound like what they are. Glycolysis, break it down. Gluconeogenesis, build it up. This is 14.4 of the textbook, incidentally. I think it's 14.4. Don't quote me. It might be slightly different in different versions, but it's definitely chapter 14. So what is it? It's the synthesis of glucose from simpler precursors, namely pyruvate, also lactate, also other things, but we'll get into that. These are not the only two things that can form glucose. Occurs mostly in the liver. We're going to start talking more and more about the liver because the liver is the glucose master of the body. It really is. It regulates glucose concentrations. It engages in glycolysis and gluconeogenesis as needed depending on what's going on. So it does use 7 of the 10 enzymes that act in glycolysis, but there are 3, count them, 3 exergonic steps in glycolysis. And those require different enzymes in gluconeogenesis to bypass those exergonic steps. That first of all, they can't operate in reverse. It's just impossible. And second of all, there is an added benefit to that insofar as gluconeogenesis and glycolysis can be regulated separately, so that the whole thing is not a waste of time. Okay, so big picture, here we see lactate being converted back to pyruvate, or if we have large concentrations of pyruvate, this can, it's showing it going through the citric acid cycle. That's primarily because there's a shared intermediate, which is oxaloacetate. From there, we go to PEP, or phosphoenol pyruvate, which is the second to last compound in the glycolytic pathway, and on up through to glucose 6-phosphate, and finally glucose. There are other players, as mentioned. There are glucogenic amino acids. Glycerol from triacylglycerols can go into the pathway at a different point. And in plants, which we're really not going to talk about very much, they have their own way of doing this, 3-phosphoglycerate after carbon dioxide fixation. Okay, so this gluconeogenic process occurs pretty much everywhere. Mainly occurs in the liver in mammals. We're not doing gluconeogenesis in our muscles, for example, at least not very much, if at all. Okay, so just to recap a little bit, lactate enters the pathway via pyruvate. We know that lactate is produced by exercising muscle, and it's also produced by red blood cells, can be reconverted to pyruvate in the liver and go into gluconeogenesis. Glycerol actually enters via DHAP, dihydroxyacetone phosphate. So the glycerol moiety of a triacylglycerol can go have a gluconeogenic fate. The liver actually takes the glycerol and converts it to DHAP in a two-step reaction, and then on up through gluconeogenesis, i.e. glycolysis in semi-reverse. And then finally, amino acids, they can become gluconeogenic by entering the pathway via pyruvate or by citric acid cycle intermediates. We haven't really talked about the citric acid cycle yet. I mentioned that oxaloacetate is one of them. An interesting thing about amino acids as a source of gluconeogenesis is this does become a major source of glucose during an extended fast. And the way that works is muscle tissue hydrolysis. So we're actually, wasting muscle. Will release glucogenic amino acids. There's a subset of amino acids that are considered to be glucogenic, just FYI. Amino acids can produce, these glucogenic amino acids can produce alpha keto acids. Well, there's ketogenic amino acids as well. And we're going to talk about ketone bodies and things. But the main point here, really, to boil it down, is that lactate, glycerol, and certain amino acids are sources for gluconeogenesis. So here is the whole shebang, gluconeogenesis and glycolysis. So as already noted, it is very important to recognize that these are not just identical pathways running in opposite reactions. And here in this figure are clearly identified the three exergonic reactions in glycolysis and the reverse strategies on the part of gluconeogenesis. And you'll notice in each case, it's different enzymes. In the reversible reactions, it's the same enzymes. And that is simply controlled by cellular concentrations. You know how much I love talking about Q, ln of Q. So it's just, that balancing act between reactant and product. If you prefer Le Chatelier, we can think of it as a Le Chatelier principle kind of thing, shifting equilibriums. So, but for these exergonic things, different ball game, different, the nature of the regulation is typically allosteric or some kind of regulatory mechanism, right? So, okay, so you have to know these. You have to know these points of regulation. Hexokinase, PFK1, pyruvate kinase are the three glycolytic enzymes that are regulated. And in reverse, it's Pyruvate Carboxylase, PEP, we're going to go through all this, PEP carboxykinase, fructose 1,6-bisphosphate, and glucose 6-phosphatase. So we're going to go through all these bypass reactions. They're called bypass reactions, by the way, because they are bypassing the glycolytic reactions that are exergonic. Okay, so as we know, these essentially irreversible glycolytic reactions are characterized by a large negative delta G. And so here they are, just to see them, right? The standard delta G is here, but under cellular conditions, it's even larger and more negative. So this is step 1 of glycolysis, catalyzed by hexokinase. This is step 3 of glycolysis, another large negative free energy catalyzed by PFK1. And this down here is the pyruvate kinase reaction that generates pyruvate, negative 16.7. It's actually, I mean, here the standard free energy is actually a bit larger than what normally happens in the cell. So everything else reversible. And that's that. These are just, this is just the numbers game here in this table. Okay, so what is this first bypass? This is the only one that actually requires 2 reactions. And potentially we could start from lactate, but always, you know, we're gonna, pyruvate is kind of like, it's the end of glycolysis, and we'll think of it as the beginning, in this case, is the beginning of gluconeogenesis. Even though we saw there are alternatives, right? We're gonna be focusing on pyruvate for now. So let's say pyruvate is already in the cytosol. And I think here's a good moment to pause, because I'm not sure that I emphasized very much that glycolysis is taking place in the cytosol. So pyruvate ultimately has to get into the mitochondria if it's going to go for the citric acid cycle, if it's going to, I don't want to make this too complicated. So anyway, at the end of glycolysis, pyruvate is in the cytosol. It does get transported into the mitochondria. Okay, so that's point 1. But here, okay, if we're doing gluconeogenesis, the first reaction is catalyzed by pyruvate decarboxylase in the cytosol. Just, I'm gonna zoom ahead. I want to check myself. Well, there are a couple possibilities. Let's put it that way. Hold your thought about where things are going down. Let's just focus for now. Forget about cytosol, forget about mitochondria. Let's just focus on the reactions, because we're going to come back to this. The first reaction is catalyzed by pyruvate decarboxylase. Another classic case of an enzyme telling you exactly what is happening, right? Because except we have a mistake. This should say pyruvate, because if it said decarboxylase, well, I make my point very well, because if it really were decarboxylase, it wouldn't be describing what was going on. It should be saying pyruvate carboxylase, because that's what's happening, okay? This is the name, this is the correct name of the enzyme. Pyruvate decarboxylase does exist, by the way. So we're taking 3-carbon pyruvate and we're making a 4-carbon oxaloacetate. So we're taking pyruvate and we're adding CO2, okay? Therefore, it consumes CO2 and it just so happens it also makes ATP. We'll be looking at it in more mechanistic detail in a moment. Then after we have oxaloacetate, there is a second enzyme that comes in. This is enzyme 1, right? And then the second reaction converts oxaloacetate to PEP. And this is, in fact, a decarboxylation. So no sooner have we carboxylated our compound pyruvate to make oxaloacetate, we're turning around and we're removing it. But of course, we're not going back to pyruvate. That would be pointless. We're going from oxaloacetate to PEP. So as you may recall, PEP is an intermediate of glycolysis, but in this case, instead of going from PEP to pyruvate, obviously we're going from PEP to, I don't know, $100 to anyone who can remember what's before PEP. I'm not sure. Oh God. Is it one? What is it? 2-phosphoglycerate. So we would go to 2-phosphoglycerate, and up and up, right? Okay. So let's move ahead here and see how this looks, because it's, I think, a little easier to visualize just to understand all this for actually looking at the reaction. So we have bicarbonate as our source of CO2, and it combines with pyruvate, and courtesy of the enzyme Pyruvate Carboxylase, ATP is consumed here. And we're going to be talking as we go forward about how gluconeogenesis is consuming things like ATP and GTP, not generating it. So we make our oxaloacetate here. And then once we have oxaloacetate, this is actually the chemistry that's happening in this next step here. This quickly turns around and does a nucleophilic attack here on GTP. So rather than consuming ATP at this point, we're consuming GTP, and we lose CO2 here. This is not a mechanism you have to know, but it's here for interest, basically, because these things are interesting. But basically, what do we need to know? We need to know that we're carboxylating pyruvate to oxaloacetate. We're consuming ATP in the process. Once we have oxaloacetate, we're consuming some GTP, and we're releasing the carbon dioxide and forming PEP. So if we are doing a bypass reaction, notice, since this is also a point of regulation, this is also an exergonic reaction to the tune of -25 kilojoules per mole. Okay, now there are two possibilities. This reaction will happen as written with the two enzymes. It can potentially happen, it can all go down in the cytosol. So I'm going to zoom ahead for a moment. None of this is showing it all happening in the cytosol. Okay, I'm going to retract that statement. What the point I'm trying to make here, and this is a tricky one to explain, I'll tell you that right now. What I'm trying to say is that there are two possibilities in terms of this bypass. If pyruvate is our precursor, we're going to get into something which I will explain momentarily as the malate shuttle. And if it's happening in this manner, which is considered to be different, there is no malate shuttle. Now, you don't know what the malate shuttle is yet, but I'll tell you right now that in the case where we have a malate shuttle, there is a reaction that is sandwiched between this and this, okay? And it's called the malate shuttle. This is a lot, but we'll get through it. Okay. If our pyruvate is in the mitochondria, we're going to add this additional layer that is known as the malate shuttle. And in the malate shuttle, which we're going to see visualized on the next slide, there are certain things that happen. Oxaloacetate is converted to malate. And then malate is exported out of the mitochondria by a membrane transporter. Now, the most important thing to recognize here is this is a redox reaction where malate is the reduced species and NADH is consumed, i.e. it's oxidized, okay? Malate reduced, I'm going to make this point again, reduced malate, it has more electrons, right, is exported out of the mitochondria by a membrane transporter. That means this is called a reducing equivalent. We have taken a reducing equivalent and we've pushed it out into the cytosol, right? What happens to malate when it is reoxidized to oxaloacetate in the cytosol is that NADH is regenerated. So now we're going to look at the figure, but think about this. Under these circumstances, this is an interesting way to move NADH into the cytosol from the mitochondria if the mitochondria has high levels of NADH. I'm going to show you the next figure and we'll talk about it again and you'll tell me if this is making any sense to you. Okay, so we have our pyruvate here, right? And it gets carboxylated in the first enzyme of the first bypass reaction of the gluconeogenic pathway, right? So that makes oxaloacetate. This in turn, as I just noted, we have the oxidation of NADH, right? And the reduction of malate. So this happens in between the pyruvate carboxylase reaction and the PEP-CK reaction or the PEP carboxykinase reaction. So what we have done in this process here, and this is a situation where we're seeing pyruvate is already in the mitochondria, okay? So this is the distinction we're making here. Then this malate shuttle will go forth. So we take the malate, a couple things happen. Pyruvate is going to go, I mean, the oxaloacetate is going to go on its merry way and go through reaction two to form PEP, the decarboxylation and the GTP consumption and all that. And in the meantime, malate will get reoxidized to oxaloacetate. I mean, this has to happen, actually. Malate has to form the oxaloacetate again, right? Because the oxaloacetate got converted to malate. So now the malate's going to go back to oxaloacetate. Then this will be the substrate for the PEP-CK reaction. And we have transferred NADH out of the mitochondria and into the cytosol, which can be a good thing if we need NADH in the cytosol. It all depends on what's happening, right? So this is kind of the moral of the story, but it's all taking place in the context of gluconeogenesis. Is that making any sense? So. Do you want to think about it for a little bit? So I'll just try again one more time, just to explain all this. Pyruvate is already in the mitochondria. We know it's a two-step reaction to get to PEP, right? So that's the big picture. We're trying to get to PEP. But this has another layer of complexity, really, because in between the pyruvate carboxylase reaction, which forms oxaloacetate, we have this malate shuttle. This is the name of this, the malate shuttle. The malate shuttle is converting oxaloacetate to malate, consuming NADH in the cytosol, in the mitochondria, regenerating it in the cytosol as malate goes back to oxaloacetate for the second reaction of the gluconeogenic bypass reaction. Okay.
Is the goal to just get oxaloacetate outside of the mitochondria? You have to
malate, really malate is being transported, is the malate shuttle, and it's a way of getting NADH from the transporter is for malate, right? Not NADH. Yeah, so the point really is to get NADH into the cytosol. Oxaloacetate, yes.
Oxaloacetate. That on its in the other diagram that you showed us, where a lactate is a precursor, the oxaloacetate can exit the mitochondria on its own in that sense.
So here's the deal. If lactate is the precursor, it's gonna be converted to pyruvate in the cytosol instead of being directly transferred into the mitochondria. So in this case, lactate is converted to pyruvate in the cytosol, right? And then in this case, we still have to transport pyruvate into the mitochondria, but there is no malate shuttle. Note, there is no malate shuttle here.
So oxaloacetate can't exit the mitochondria on its own. It has to either exit as malate or PEP.
Correct. Oxaloacetate is retained. That's a good point. Is retained in the mitochondria here, and in this case over here, it's converted to malate is transported out. Anyway, bottom line is, if we're going from lactate to pyruvate, no malate shuttle, but if pyruvate is being directly transported into the mitochondria as pyruvate, we go through this malate shuttle thing. Yes?
So the pyruvate on the left would be like the pyruvate fresh out of glycolysis, right?
Correct.
Okay.
Correct. So, but what really dictates these pathways? I'm not. What's important here? What's going to dictate these pathways is the relative, as you point out. Are we fresh from glycolysis, or have we gone through lactate? What are the prevailing concentrations of pyruvate versus lactate versus cytosolic requirements for NADH is going to control what happens here. Okay, so now we can take sort of a more cursory look at the second, not too cursory, but a more abbreviated look at the second and third bypass reactions. Again, both strongly exergonic. They're both dephosphorylation reactions. However, note that we are not transferring these phosphoryl groups that are coming off our substrates to ADP. They're being released as inorganic phosphates. So fructose 1,6 bisphosphatase, or if you prefer, FB-PACE1, converts fructose 1,6 bisphosphate to fructose 6 phosphate. Another $100, but I know the answer to this one. What is this, the reverse reaction of? What is the glycolytic reaction here? Fructose 1,6, what enzyme catalyzes the glycolytic reaction? Very good. PFK1 catalyzes the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. I don't have $100 on me right now, but see me next class. So FB-PACE1 is the reverse of PFK1 as we have it in glycolysis. But now we're taking fructose 1,6 bisphosphate and we're dephosphorylating it to fructose 6 phosphate and we're releasing inorganic phosphate, okay? Glucose 6 phosphatase, hydrolysis of glucose 6 phosphate to glucose. $5. The price is going down. What's this the reverse of? Hexokinase, right? Both exergonic, right? But we're not getting a payoff here. We're releasing phosphates, and that's energetically favorable, but we're not getting the payoff of forming any ATP from ADP. Which leads us to this. Recall in that first bypass we consumed for the Pyruvate Carboxylase and PEP carboxykinase reactions, we consumed an ATP and a GDP ×2, right? Because everything is ×2. So we consumed where's the other ATP coming? So for, back it up. Let me think here. Going back, doing a little accounting. This is 4. This is 4, right? So let's do the accounting together. trying to figure out, I feel like I've missed something here. We're consuming 4, but why only 2 GTP? Oh, wait, no, this is, I think I get it. Okay. Shouldn't say 4 here, I should say 2. This is 2 ATP here, but where else? Let's find out where else are we consuming ATP here? This one, okay. So here's another 2 ATP. This is a reversible reaction, right? This is not an exergonic reaction. So, and then of course, what else? Where are we consuming our NADH? Here we're consuming 2 NADH. And so that's the cost. This is the cost, right, of gluconeogenesis, and that's pretty significant. But we're getting a big, we're getting glucose, right? We're getting a nice source of energy that, okay. So what else? We can think about now a little bit about some of these other sources, which are glucogenic amino acids as precursors to gluconeogenesis. So glucogenic means what it sounds like, which is these are amino acids that can undergo a net conversion to glucose through intermediates. So they're pretty much all amino acids are glucogenic except lysine and leucine. Let's just move on to this. Well, no, let me talk about this a little bit more. Okay, so these are all the glucogenic amino acids grouped by their site of entry. So we can pass through different things, and pyruvate, of course, we recognize. So alanine, cysteine, and so forth here can enter the glucogenic pathway through pyruvate. Others can only, well, not only, I should say, there is duplication here, but duplication? Actually, no. So other glucogenic amino acids enter through alpha ketoglutarate, succinyl CoA, fumarate, and oxaloacetate, which are all intermediates of, you guessed it, the citric acid cycle. You don't have to memorize this. This is just something to know. They enter through different glucogenic amino acids, enter through different intermediates. Now, lysine and leucine are something called ketogenic amino acids. I think the keto diet is sort of like probably, it was a real fad there for a while. I think we hear about it less these days, but for, I don't know, I'm sure there are some people out there who are still like pursuing. Are you familiar with the keto diet? No? Yes? So what do you know about it?
It's super low carb.
Super low carb, a lot of proteins and a lot of fats. So without carbs, what are we not getting? Glucose, right? So what are we going to have to do? We're either going to have to break down, we're going to have to use our amino acids that are coming through our diet. And this is one way to get to glucose, but we're going to talk later about ketone bodies, and we're going to see what the chemistry of that is. We could also potentially get some glucose through fats, but that's really not a typical pathway for gluconeogenesis. Glycerol is really the only thing, right, that's gonna give us, and that's not a very large, significant part of the lipid molecule, right? It's just one small moiety. So There are some amino acids that are both glucogenic and ketogenic. So the key here is anything in purple can be both. Anything in red is strictly glucogenic, and anything in green, which is just lysine and leucine, are ketogenic. Again, no need to memorize this, but this is a good figure. It like really shows you, you know, how things enter and so forth. Let's see, does it really line up with, yeah, fumarate, phenylalanine. There are some discrepancies here if we compare closely, because this says fumarate is only phenylalanine and tyrosine. This one, oh, but aspartate is glucogenic. Yeah. So interestingly, they show aspartate here is entering through oxaloacetate. They show aspartate in both places here, entering through fumate, and so, you know, one of them's right. Broad strokes is fine for this. Okay, even though it's a little early, I feel like now is a good time. Let's take a 10-minute break, and then if we need another break, we'll take a second break. I'll give you your exams back. You can come and get them. I don't see Ego. Oscar? Javier? Tamara, are you there? Tracy? Rosales? Rosalise? Yeah. Kishab? Sarah? Shamia? Emanuela? You're welcome, Cynthia. Alexander.
Thank you.
You're welcome. So 10 minutes, look it over. Let me know if you have any questions. We can go through the answers if you want, but I'm going to keep lecturing. Maybe we'll reserve a little time at the end for. Okay, so I already noted that if these were perfectly reciprocal pathways, we'd have a not very good situation with respect to regulating them because, yeah. Oh, I forgot. Okay. Or is it not? Okay, come on. I think I'm going to have to restart via. Ain't cooperating. Okay, so as noted, if these pathways were just the reverse of one another, we would have something called a feudal cycle. We wouldn't really be able to accomplish much in the way of chemical and biological work. And we have a solution through something called reciprocal regulation. This means we can halt one pathway while activating the other, or activate the one by halting, vice versa, in other words. So we're going to see that our typical regulatory mechanisms act on the enzymes that are unique to each pathway, those exergonic reactions. So those are going to be our focus. Although before we really get to the reciprocal regulation of glycolysis, gluconeogenesis, we're going to take a look at hexokinase regulation and how it differs in tissues like the muscle and tissues like the liver. Because we have these things called isozymes, and isozymes are enzymes that catalyze the same reaction, but they're slightly different proteins. They differ in terms of things like kinetics, their substrate affinity, the way they're regulated, and so forth. So as we know, hexokinase is the first step of glycolysis. It phosphorylates glucose and makes it glucose 6-phosphate. So there are 3 different isozymes that operate primarily in the muscle: hexokinase 1, 2, and 3. We're not going to concern ourselves particularly with the differences among those 3 enzymes, but we are going to note that they are all allosterically inhibited by their product, which is glucose 6-phosphate. Hexokinase 4, on the other hand, sometimes commonly known as glucokinase, is not, interestingly enough, inhibited by glucose 6-phosphate. Why? Well, this has to do with the role of the liver in responding to the availability of glucose in the body and rectifying concentrations of glucose in the body as needed. So the liver is really major in this regard, and it's kind of interesting how this is achieved. So back to our friend Michaelis Menten, we're going to see a real biological example here of why things like Km matter, right? So glucose concentration and enzyme activity, if we compare hexokinase 4 and hexokinase 1, produce very different graphs here. And note that hexokinase 4, which operates in the liver, is half saturated around 10 millimolar. This is a much, much higher concentration than half saturation for hexokinase 1, for example. So low Km, high affinity for hexokinase 1, high Km, and not just the affinity, but changing affinity, right? We come back to this sigmoidal kind of graph, and we can see that hexokinase 4 would be capable of responding to changing glucose concentrations, much more so than this very high affinity Km, which is just going to bind as much glucose as quickly as possible, and then it's going to not really respond in any particular way as you head towards saturating concentrations. So there's a couple things going on in the liver. One is that GLUT2 is a transporter for liver cells that is very efficient and equilibrates quickly, meaning that it is highly responsible to changes in physiological glucose concentrations. So, so that means a couple things. One is that there's more capability in the liver to regulate the activity of hexokinase 4 simply by changing glucose concentrations, right? Because you can see that, if we're a very high, well, let's look in here. If we're at 5 millimolar, we're ramping up pretty quickly, right? And then it starts to level off a little bit around 10 millimolar. So different responses by the enzyme according to different glucose concentrations. Hexokinase 1, on the other hand, as already pointed out, can't do that. But there's more. There's more. So there is a regulatory mechanism for hexokinase 4 that is a bit different from the mechanisms that we've looked at previously for regulating the activity of hexokinase 4. So there is an actual regulatory protein that binds to hexokinase 4. And when this happens, hexokinase 4 with the regulatory protein gets sequestered here in the nucleus. So when does this happen? Well, it happens in response to high levels of fructose 6 phosphate, which activates enzyme sequestration by this process of, it leads to the very tight binding of the regulatory protein to hexokinase 4. On the other hand, when there are high levels of glucose, so glucose and fructose 6 phosphate are actually competing for the allosteric site. When there's lots of fructose 6 phosphate, it's binding where glucose normally would in the hexokinase enzyme. And when there's a lot of this bound, we have this situation. But when there's a lot of glucose bound, outcompeting any fructose 6 phosphate for the allosteric site, no, the regulatory protein dissociates, and we have hexokinase 4 free to pursue its activity. Okay, so that's a little different, but this is not, we're not really talking about reciprocal regulation, right? Which is what I sort of kicked this whole show off with. So now we're going to talk about that. And we're going to look at PFK1 regulation by ATP and citrate to understand how PFK itself might be allosterically controlled. So this is a situation where ATP inhibits PFK1 allosterically, and ADP and AMP also bind to an allosteric site and relieve the inhibition, i.e. promote the activity of PFK1. Does this make sense? Let's think about this for a moment. Does it make sense to have ATP inhibit PFK1? Why or why not? What do you think? What would ATP, a lot of high concentrations of ATP signal?
Enough energy.
Plenty of energy, right? Do we need to be going forward with glycolysis if we have large amounts of energy at our disposal? No, I mean, ATP is a product of glycolysis, right? So on the other hand, if we need to be generating ATP, high concentrations of ADP and AMP would indicate that we need to get going and start producing more ATP. So okay, so AMP and ADP, if you look at this down here, stimulatory, ATP inhibitory. But there's more. There is citrate, which we're going to be seeing in the citric acid cycle. Also an allosteric regulator, i.e. inhibitory in this case of PFK1. So high citrate also means the cell is meeting its current energy needs. So inhibitory, and of course, ATP, we already discussed inhibitory. Here's another compound, though, that we see as an activator, fructose 6 bisphosphate. And this is something we haven't seen before. You might think we've seen it before because it would be easy enough to confuse with fructose 1,6 bisphosphate, but it ain't the same thing, not at all. Its main purpose in life is to regulate glycolysis, and we'll be expanding on that very soon. But it is, let's keep in mind, fructose 2,6 bisphosphate activated. Okay, so just to look at the PFK molecule, which is like a lot of these enzymes, is a multi-subunit, pretty large protein. You know, I think it's always tempting to think of the substrate as big and the enzyme is small. I don't know if you ever think that way, but it's the complete opposite, right? These enzymes are these mega things, and the substrates typically are relatively speaking, quite small. So plenty of binding sites here for things like ATP, as we said, activating, ADP, inhibitory. We can also see that there are binding sites for fructose 1,6 bisphosphate, which we haven't really talked about. So we're just gonna, that's okay. We're gonna keep the focus on our allosteric regulators, ATP and ADP. Okay, so if PFK1 is the forward reaction for glycolysis, well, I shouldn't really say forward. PFK1 we've just been discussing, right, is activated when we have a signal for low energy stores and inhibited when we're talking about high energy stores. Now, what about FB-PACE1? Well, this is doing what? This is taking fructose 1,6 bisphosphate and it's converting it to fructose 6 phosphate. This is the final, this is the third bypass reaction for gluconeogenesis, right? So it, on the other hand, would be inhibited by AMP, because if we have low energy, we're not in the business of making glucose, right? We're in the business of breaking, we're in the business of activating PFK1. Now, I don't really love this figure because I think at the least they should be showing high stores of ATP as being activating to FB-PACE1, but that's okay. You get the general idea, right? But this is a figure directly from the book. I think I honestly don't have an explanation for you for why they don't at a minimum show ATP as being activating to FB-PACE1, because it should be. Just kind of follow the logic in terms of energy stores and what you want to do. High energy, you're probably going to be activating gluconeogenesis. Low energy, you're going to be activating glycolysis. Okay, so now we're going to talk about fructose 2,6 bisphosphate, which is complex but fascinating because it involves a whole bunch of things that we're going to start off with the simplest level, and then we're going to progress from there. So it activates PFK1, it inhibits FB-PACE1. So how is it controlled? That is what's gonna introduce kind of the layers of complexity here. It is under the control of hormones. And hormones are not just things like estrogen and testosterone and some of the things we looked at before. Insulin and glucagon. I don't know if you think of insulin as a hormone, but it is. So we have insulin So I'm going to get to this on the next slide. So I don't even know if I want to say it yet. You know, I think it's fine. We can leave it. I'll be picking up with what insulin does and what glucagon does on the next slide. So just keep in mind for now that fructose 2,6 bisphosphate activates PFK1, inhibits FB-PACE2. Glucagon, well, no, I'm not talking about hormones yet. Okay, so next. Now I will talk about hormones. Insulin is released when blood glucose level is high. And in order to understand the production of F26B itself, so we want to understand what controls the concentration levels of F26BP, right? That's the goal of what we're going to be talking about here. And if we have lots of insulin, there is an enzyme that controls levels of this compound, right? And that enzyme can be considered to have like two portions. The PFK2 portion, it is a single bifunctional protein, okay? It has the PFK2, not PFK1, PFK2 portion, and the FB-PACE2 portion, not FB-PACE1, not PFK1, okay? So if we have a lot of insulin, this kicks off a phosphoprotein phosphatase function. Okay, so what does that mean? That means it dephosphorylates this bifunctional enzyme. In the dephosphorylated state, this enzyme is active. If it is active, PFK2 predominates, and we raise the concentration of fructose 2,6 bisphosphate. I'm going to just give you a moment to absorb that because it's sort of a lot to take in, right? So phosphatase activity dephosphorylates bifunctional enzyme, PFK2 becomes active, raise concentration of this compound. What does this compound do? It stimulates glycolysis. Okay, at high levels. All right, now what? What if it's the other way around? Now we have high levels of glucagon, a different hormone. And it kicks off the activity of a protein kinase, more specifically C-AMP dependent protein kinase. And it uses ATP to phosphorylate our bifunctional enzyme. When phosphorylated, this bifunctional enzyme, note the presence of a phosphoryl group here, right? And it's a, a particular serine residue, but whatever. Now FB-PACE2 is active. So the weird thing here, in a way, is that phosphorylation of one enzyme promotes the dephosphorylating activity of this bifunctional enzyme. So when FB-PACE2 is active, that lowers the concentration of F26BP. What does that do? It inhibits glycolysis because high levels of this provoke glycolysis, low levels of it inhibit glycolysis. Good. FB pace activity lowers F26B P, glycolysis inhibited. This is the same game all over again. I think I have kind of made it clear, so I don't think I need to go over this. I just presented it two different ways because this slide has a little bit more information. So maybe when you're, or a slightly different way of looking at it. But it's the same thing, okay? So I don't think I need to go over it all over again. It's a slightly closer look. Okay. What about pyruvate kinase? What does pyruvate kinase do? Who remembers? I remember. Even though I have a much more aged brain than you guys. What does pyruvate kinase do?
Shut the last enzyme.
It is the last enzyme in the glycolytic pathway. So let's do a little exercise here. I'm going to let you guys maybe try to parse this figure for a minute. What's happening here? We want to think about when pyruvate kinase is activated and why or why not, depending on the tissues, right? Let's focus on the liver first. Who wants to take a stab at explaining in words what's going on here? In the presence of glucagon, let's say.
Pyruvate kinase is not reversible, so because the gluconic gets activated.
How is it? Is it? Okay, so what happens here? Glucagon activates, be careful, glucagon activates pKa. What does pKa do? What is pKa doing here? Okay, and when correct, and then what does that do to pyruvate kinase? It inactivates it. So glucagon is in effect doing what? Inactivating pyruvate kinase. Now, on the other hand, if pyruvate PP, so pKa, just to be clear here, because there's abbreviations, right? This is pyruvate kinase A. And it is it is as a kinase operating, as Alexander pointed out, to phosphorylate this enzyme. Every enzyme we're going to look at that's regulated through phosphorylation, it's very important to keep in mind that phosphorylation can be activating for some enzymes and inactivating for other enzymes. It's totally case dependent. We're going to see a lot of cases. So far, weirdly, I would say more often than not, phosphorylation is activating, but we've seen a couple examples so far where pyruvate kinase is, where phosphorylation is inactivating. But it just depends. Honestly, it just depends. So in the cases of dephosphorylation, our pyruvate kinase is now activated, but notice it's glucagon and insulin are acting through the liver, okay? They don't, for other glycolytic tissues, we're back to regulation through other means, which is what is activating here? Well, fructose 1,6-bisphosphate is activating. Right. And things like ATP, indicating high energy levels, acetyl CoA, and long chain fatty acids, all symbols of high energy conditions, are inactivating to this enzyme. Okay. Moving right along. Okay, so let's consider what happens if we have a plentiful store of fatty acids. Well, we haven't talked about the breakdown of fatty acids yet, but they do produce, the breakdown of fatty acids produces acetyl CoA, okay? So now we're looking at the first reaction of gluconeogenesis, right? And also the reaction of this, we're putting, unfortunately, I'm putting the cart before the horse a little bit with this slide because we haven't talked about the PDH complex. For those of you who have had a little, anyone encountered the PDH complex? Okay, so I'll explain it right now, but this is going to be a little confusing until you know more. You certainly understand, hopefully, you understand at this point that pyruvate carboxylase is one of the enzymes that is involved in the conversion of it is the enzyme involved in the conversion of pyruvate to oxaloacetate and then oxaloacetate to PEP with the second enzyme, right? PEP carboxykinase. So the PDH complex actually takes, well, you can see it right here. The PDH complex, which we'll be talking about very soon, takes pyruvate and converts it to acetyl CoA. And then acetyl CoA goes on into the citric acid cycle. So acetyl CoA, if there's plenty of it, it stimulates pyruvate carboxylase because we're saying, let's go gluconeogenesis. But it inhibits the PDH complex because that's saying, let's break acetyl CoA, let's make more acetyl CoA and go ahead with the citric acid cycle, which is energy producing, right? So the logic is always there. Plentiful energy stores stimulate synthesis, hold off on, stimulate anabolism, right? The buildup of energy-rich compounds, slow down on catabolism, which includes not just glycolysis, but things like the citric acid cycle. Okay, finally, we are concerned as well, although I'm going to give this pretty much the short shrift because we're not really talking, as you know, a lot about DNA and transcription in this course. But on a longer term basis, we've been really talking about allosteric modification. On a longer term basis, we can have transcriptional regulation of these enzymes. And if we have high levels of insulin, we're going to increase expression of catabolic enzymes. And if we have the opposite situation, lots of glucagon floating around, the opposite could be true, right? But all these enzymes, some of which we've talked about, some of which we have not, are all involved in glycolysis, production of acetyl CoA, and so on and so forth. So we either have increased expression, right, or decreased expression. And that's it for the pentose phosphate pathway. Oh my God, I forgot that was in these slides. Okay, we'll start in on that. I hope I'm staying on schedule here. I think I am. So, pentose phosphate pathway is an alternative pathway for glucose oxidation. Glucose has many fates in the cell. And remember when we talked about glycolysis, we said there's a committed step, right? The committed step is PFK1, I do believe. Before that, glucose can get phosphorylated, but can go to other pathways. In particular, we're going to look at the overall pathway for the pentose phosphate pathway and see what happens here and why it happens. So why does it happen? Well, rapidly dividing cells need certain compounds, right? Bone marrow, skin, intestinal mucosa are all rapidly dividing, and unfortunately also those of tumors. So all of these rapidly dividing cells need pentose, the pentose, ribose 5-phosphate to make not only RNA and DNA, but things like our friends ATP, NADH, FADH2, and coenzyme A, all super important, right? That's one possibility. The other possibility in other tissues, not necessarily rapidly dividing, the essential product of the pentose phosphate pathway is the electron donor NADPH. So this is needed for biosynthesis that depends on reduction of NADPH, or actually, interestingly, to counter the damaging effect of oxygen radicals. So overall pathway, glucose 6-phosphate combines with NADP+, got some water thrown in. We form ribulose 5-phosphate that can ultimately be converted to ribose 5-phosphate or not. And we get our NADPH, right? As well as some carbon dioxide and hydrogen ion. So obviously this is very important. We need glucose for energy needs, but we'd be in a pickle, in a lactic acid pickle fermentation, if we weren't able to make things like ribose phosphate, if we weren't able to do this as well, right? Okay, so moral of the story, pentose phosphate pathway directs glucose 6-phosphate to other catabolic fates, giving us these specialized products we just talked about that are needed by the cell, i.e. pentose phosphates and NADPH. It has an oxidative phase and it has a non-oxidative phase. The non-oxidative phase will recycle. Remember, the overall reaction showed the product is ribulose 5-phosphate, right? That can be recycled in the non-oxidative phase. This will happen, this recycling will happen if we just need NADPH, right? If we need these if we need ribose 5-phosphate, for example, this can continue on to make ribose 5-phosphate, and the ultimate fate of that is for nucleotides and coenzymes and so forth. Okay. So that's the big picture. We can, in terms of the pathway here, there's obviously more to it than just what I spoke about. But this is the big picture, right? So let's look at the first couple steps of the oxidative phase, where we produce a couple compounds here, 6-phospho. You don't have to know all these intermediates, by the way, I don't really care. But this is what occurs, right? So let's relate this back to what we saw in the previous figure. We have two enzymes at play here, glucose 6-phosphate dehydrogenase and lactinase. The ultimate thing that happens is we get to here, right? In a couple steps, a couple enzymes to get from glucose 6-phosphate to 6-phosphogluconate. That's the oxidative phase, the first two steps. Then we have two more enzyme catalyzed reactions, the third and fourth oxidative steps. And this is where we get a bit of a more interesting things happening in terms of what we want to get out of this reaction. We get oxidation and decarboxylation of our 6-phosphogluconate product to form ribulose 5-phosphate and, importantly, NADPH. This, as noted, can go on to form ribose 5-phosphate. At this point, after 4 reactions in the oxidative phase, 1, 2, 3, 4, we have generated 2 NADPH and a carbon dioxide and a ribose 5-phosphate if we continue on, right? So notice that ribulose 5-phosphate is getting to ribose 5-phosphate is an isomerization reaction. We're just, what are we doing? We're, yeah, we got a double bond is, so we have a reduction. Have a reduction? Yeah, it looks. No, that's a reduction. Whenever we add a hydrogen, it's a reduction. So, okay, whatever. Huh?
I mean, but you're moving the other hydrogen.
Oh, that's true. You're right. You're right. So it is simply an isomerization because this is, yeah, it's not a reduction. Because I was like, what's getting oxidized? But you're right. It's merely moving the double bond up to here. Okay. And here we see it again. Oh, no, we don't see it again. In the non-oxidative phase. So I like to keep referring back to the figure here, just so we kind of keep track of our P's and Q's, or we mind our P's and Q's, whatever you do with P's and Q's. Again, I'm going to point out that instead of this, we can go around this way, right? And this depends on a couple steps. Once again, certainly not going to hold you responsible for all of these different enzymes and compounds, just the more of the broad strokes. But it's sort of beholden upon me to talk about them, right? So we have an epimerase reaction where we go from ribulose to xylulose. Okay, so this is where things start to get really interesting, but it being, and this is extremely cool, and I like to call this biochemistry on acid. I really do, because it is the most incredible thing of, if anyone can come up with a reasonable explanation for me as to why it works this way, I mean, it's fascinating, but it's insane. So we will save that fascinating insanity for our next class, which is Wednesday. And we're going to look at all the rearrangements and recycling. This is like, that's really what this is. A bunch of moving of groups around to convert glucose 6-phosphate ultimately to 6-CO2, ultimately. And we're going to see how these enzymes, transketolase and transaldolase, pull this one off. So yeah, if you have a time to take a look at this before the next class, fine. If not, also okay. But all right, here's my question in the last 2 minutes of this class. Are we on schedule? Let's take a look at the syllabus. Oops, whoops, oops. Announcement, quick, get away from this. I don't want to show this. Damn it. Okay, syllabus. Say what? I know, which is very exciting. But you never know. And yeah, so a week from tomorrow, you have your next test. Oh, I think I'm in, yeah, no, I'm in the right place. Is that right? A week from tomorrow? Yeah, a week from Wednesday. Yeah, not tomorrow. Tomorrow's the day off. So, what is today? Oh yeah, we're in good shape. I don't have the pentose phosphate pathway on here. So we'll finish up with the pentose phosphate pathway and we'll move on to glycogen metabolism. We're in good shape. But it's never too soon to do what? It's a lot of information, right? Flashcards, like I highly recommend at this stage of the game. I'm not going to sit around and just necessarily ask you about, you know, steps and enzymes, but you do need to memorize glycolysis, you know, all the steps, all the enzymes and everything. So you might note taking and, you know, testing yourself and stuff are pretty critical at this. You may have been doing that all along, but I think it's more critical than ever for this.